All right, today we're going microscopic to look at the two fundamental ways our cells make more of themselves, mitosis and meiosis. Now, I know they sound super similar, but trust me, the difference between them is, well, it's basically the difference between healing a paper cut and creating a whole new life.
So it all begs a really simple question, right? If the goal is just to make more cells, why does your body have two completely different ways to do it? Why complicate things? Well, the answer to that is the key to understanding, well, pretty much everything, from how you heal a simple cut to how life itself evolves.
And here's the answer. It's because our cells have two totally different missions. The first mission is all about growth and repair, making exact copies to keep your body running. But the second mission, that's about reproduction. It's about creating genetic variety for the next generation. So you've got two missions, and that means you need two different strategies. Okay, so before we jump into the how, the real nitty-gritty of cell division, let's spend a minute on the why, because if you understand the mission, the method makes so much more sense. All right, before we can go any further, we have to meet the key players. So you start with what's called a homologous pair of chromosomes. You get one from each parent, right? And they carry genes for the same traits. Now, before a cell divides, it copies its DNA, and that's what creates these classic X shapes you see here. Each half of that X is a perfect identical copy, and those two halves together are called sister chromatids. Seriously, getting the difference between a homologous pair and identical sisters is probably the most important thing to get right now. It's the key to everything else we're about to talk about. Okay, let's kick things off with mitosis. And honestly, the easiest way to think about this process is to picture it as your body's personal cloning machine, or a super high-quality photocopier. Its only job is to churn out perfect, identical copies. So where does this cloning happen? In what are called our somatic cells. And that's basically every cell in your body that isn't a reproductive cell. We're talking skin cells, liver cells, bone cells. Anytime you grow or need to repair some damage, these are the cells doing the work, and they're using mitosis to do it. And the whole thing unfolds like this perfectly choreographed four-act play. You've got prophase, metaphase, anaphase, and telophase. Just think of it as the cell following a super precise script to make sure it creates a perfect clone every single time. But the real critical action, the main event really, happens right here in anaphase. You have these things called spindle fibers that act like tiny little ropes. They latch onto the chromosomes and just pull. They yank those identical sister chromatids apart, dragging one to each side of the cell. This is how the cell guarantees that both of the new cells get a complete and absolutely perfect set of genetic instructions. Okay, so if mitosis is that perfect, reliable photocopier, then meiosis is the cell's genetic slot machine. Its entire purpose is to just shuffle the deck and create as much randomness and diversity as it possibly can. This right here is the engine of variation. And this whole process, this genetic lottery, is reserved exclusively for our germ cells. The cells that eventually become sperm and eggs. The goal here is the exact opposite of mitosis. It's not to make clones. It's to create cells that are totally 100% genetically unique. So how does meiosis pull this off? It has a few special tricks up its sleeve. First, it goes through 2 full rounds of division, not just one. And second, it uses these 2 really brilliant tricks to shuffle the genes, something called crossing over and another called independent assortment. Let's zoom in on that crossing over thing for a second. In the very first phase of meiosis, the homologous chromosomes get cozy. They pair up, and then they literally swap pieces of their DNA. It's incredible. They're basically trading genetic information, mixing up the maternal and paternal genes to create brand new combinations that have never existed before. And all of this brings us to the climax, the real heart of the story for both mitosis and meiosis. And that's the moment of separation, which happens in a phase called anaphase. It's in this one single step where the massive difference between these two processes becomes crystal clear. It really all boils down to this one idea. The crucial difference isn't about the names of the phases, it's about what gets pulled apart. That one little detail, that single action, changes absolutely everything. Okay, let's look at this side by side, because this is the whole game right here. In that first division of meiosis, what we call anaphase I, the cell pulls apart the homologous chromosomes. See? The X's are still intact, the sister chromatids stay together. In mitosis, and also in the second division of meiosis, the cell does something different. It pulls apart the sister chromatids. It splits the X's. So, check this out. This is what's happening just before that first big split in meiosis. The homologous pairs line up together, right down the middle. So when they get pulled apart, each new cell gets one whole chromosome from the pair, still with its sister chromatid attached. Now look at the complete contrast in mitosis. See, here the chromosomes line up single file. The goal isn't to separate pairs. The goal is to split those identical sister chromatids right down the middle. It is a totally different move, and that's why you get a totally different result.
So, after all that pulling and splitting, what's the final score? What do these two processes actually give us at the end of the day? Let's check the final tally.
And just as a quick reminder, for us humans, both processes start at the exact same place. One single diploid cell, which has 46 chromosomes all arranged in 23 pairs. This really puts it all in one place. You can see mitosis happens in our regular body cells. It splits once and gives you two identical daughter cells with a full set of chromosomes, perfect for growth and repair. Then you have meiosis, happening only in reproductive cells. It splits twice, gives you 4 daughter cells, and every single one is genetically unique with only half the chromosomes. One makes clones, the other makes variety. That's the whole story. So yeah, the bottom line is really this simple. Mitosis ends with 2 identical cells, each with the full 46 chromosomes, perfect clones. Meiosis, on the other hand, ends with 4 unique cells, each with only half the chromosomes, just 23. One is for copying, the other is for creating. And this process, meiosis, this incredible engine of diversity, it's what drives evolution. It creates the variation that allows life to adapt and change over time. But, a dance this complicated, this intricate, it's a high-stakes game. So it leaves you with a pretty big question. What happens when it goes wrong? What are the consequences when this great cellular divide doesn't go according to plan?